Hello everyone. In this lecture we will discuss the final step in the anaerobic conversion process, the production of methane. The final step in anaerobic digestion is called methanogenesis. In this step, the final intermediates, merely acetate and hydrogen CO2, are converted to methane gas by methanogens. Methanogens belong to the domain of archaea and differ in many ways from bacteria. In fact, they belong to the oldest inhabitants of our planet, having a history of over 3.5 billion years. They are characterized by typical physiological and biochemical features and are often found in extreme environments. Nonetheless, they are very common, and every anaerobic high-rate reactor producing methane is full of them. Are acetate and hydrogen CO2 the only methanogenic substrates? Well, not the only ones, but they are the most important ones. On average, about 70% of the COD conversion is channeled via acetate to methane gas, and about 30% is channeled via hydrogen CO2. During methanogenesis, the acetate molecule is in fact split into an acetoclastic reaction into a methyl group and is further reduced to methane, and a carboxyl group that is further oxidized to bicarbonate of CO2. The table lists various methanogenic substrates, together with their standard Gibbs free energy values. In addition to acetate and hydrogen CO2, other common substrates are formate, carbon monoxide, methanol, methylamines. For all substrates, the Gibbs free energy change is negative, meaning we are dealing with exothermic reactions. However, for most important substrate, acetate, the value is only minus 32.5 kJ per mole, which is very low. In most cases, the actual energy change is even not enough for creating 1 ATP, the universal biochemical battery. Interestingly, the methanogens have the ability to store the produced energy of one reaction over the membrane, and then, with the next reaction, sufficient energy is produced to form 1 ATP. The two major methanogenic substrates, acetate and hydrogen CO2, can also be transformed to each other in the so-called homoacetogenic conversion reaction. Under standard conditions, centrotrophic acetate oxidation is endothermic and not feasible. However, under specific conditions, these reactions can become of great importance. For instance, under thermophilic conditions, homoacetogenesis might become the most important conversion pathway for acetate. This then will result in hydrogenotrophic methanogenesis as the sole route for methane formation and thus COD removal. Similar observations have been done in anaerobic reactors that were exposed to extremely high ammonium concentrations. Apparently, when acetoclastic methanogens become affected, centrotrophic acetate oxidation becomes responsible for soluble COD conversion to hydrogen CO2, the methanogenic precursor for hydrogenotrophic methanogens. The anaerobic reactor is full of interesting life forms, which are able to survive and grow at the thermodynamic minimum of existence. Moreover, the little energy that is generated during the mineralization process has to be shared between the different species, which are dependent on each other. Only in a balanced microbial consortium, all reactions are able to proceed. Methanogens have a very narrow substrate spectrum, and the growth rate is linked to the amount of energy set free during the methanogenic reaction. This energy dissipation is different for acetate consumers and hydrogen CO2 consumers. Hence, both types of methanogens have different kinetic characteristics. In this table, the listed growth rates of μmax of the hydrogenotrophic methanogens are a factor 4 to 20 higher compared to the acetate consuming methanogens. In addition to the μmax, also the doubling time, the TD, and the substrate affinity constants, the Ks, is given. The table also lists the kinetic parameters of propionate and butyrate oxidizers. With regard to the acetate consuming methanogens, two sets of kinetic parameters are given, which belong to two different species of methanogens. The slow-growing acetoclastic methanogens that is characterized by a high substrate affinity, or low Ks value, belongs to the Metanoceata species. And the fast-growing organisms, characterized by a low substrate affinity and a high specific growth rate, belongs to the Metanosuccinus species. The substrate-dependent growth curves are following monokinetics. From Monod equations follows when the substrate concentration equals the Ks value, the actual growth rate is half mu max. Now let's have a closer look to the kinetic parameters of these two methanogens. Metanosaeta is characterized by a low value of mu max, so they are relatively slow growers, but with a high substrate affinity, indicated by a low Ks value. So when this bacterium dominates our sludge, the sludge growth is rather slow, but the attainable effluent acetate concentration is very low, which is advantageous. The Metanosacinus species grow much faster, but the substrate affinity is much lower, as indicated by the high Ks value. The latter will result in more residual acetate in the effluent. It must be noted that when acetate concentrations are below the Ks value of Metanosacina, the Metanosaeta species grow faster. Based on these kinetic considerations, we rather would select for Metanosaeta species in our methanogenic sludge, as they have a high substrate affinity and thus can attain very low effluent VFA concentrations. For a wastewater treatment system, these are very important features. In addition to these kinetic differences, there are also biochemical and morphological differences between these two methanogens, explaining why Metanosaeta is generally found in all methanogenic sludge granules around the world. The number of methanogenic bacteria inside the sludge granule largely depends on the substrate. Feeding the sludge only with acetate will result in an enrichment of the acetotrophic methanogens. However, when feeding with more complex substrates, then also acidifiers and acetogens will constitute the sludge granules. On average, one may estimate that about 10 to 50% of the bacteria present are methanogen-consuming acetate and hydrogen CO2 consumers. The slow-growing Metanosaeta species is most commonly found in methanogenic granular sludge. It is characterized by elongated filaments. They have a hydrophobic surface with an electrophoretic mobility of about 0 at pH 7, so they stick together. Metanosaeta can only consume acetate, which is split into a methyl group that is further reduced to methane, and a carboxylic group that is further oxidized to CO2. This spaghetti type of organism easily forms clumps that constitute the core of the granular sludge. The fast-growing Metanosacinus species has a coccoid, grape-like structure and is bound together with excreted extracellular polymeric substances. Metanosacinus is more versatile in substrate consumption and is able to convert both acetate and hydrogen CO2 to methane. Some species are even able to convert methanol and methylamines. Now, if you start up a reactor, we automatically select for Metanosaeta species as long as we keep the effluent acetate concentration to a low level. At these levels, Metanosaeta grows faster than the Metanosacinus species. Moreover, Metanosaeta forms a granular sludge with a diameter of 2 to 5 mm and high settleability. Metanosacina, on the other hand, forms a lighter coccoid cluster and is more prone to washout when the upward velocities are increasing along the reactor startup. Hence, Metanosaeta are the species we should focus on during the startup. A predominance of these species ensures low effluent acetate concentrations and the growth of well-settleable granular sludge. As shown in the previous table, Metanosaeta species are characterized by the lowest growth rate of all the trophic groups involved in the anaerobic conversion process. Hence, particularly during the startup, reactor operation and increments in loading rates is governed by the growth and development of a stable acetoclastic methanogenic sludge bed, consisting of Metanosaeta species. The formation of anaerobic granular sludge is a common phenomenon in anaerobic high-rate reactors. The exact composition and structure is determined by the wastewater composition. Based on the difference in kinetic properties of the different trophic groups, a layered structure of the sludge granules can be expected. And the here cited paper gives a schematic presentation how such layering may evolve. When feeding with carbohydrate-containing wastewaters, the acidifiers will proliferate at the outside of the granule, since they have the highest growth rate and the highest yield of all trophic groups. The intermediate products of the carbohydrate acidifying bacteria will diffuse into the granule to be subsequently converted by the acetogenic bacteria to acetate. And finally, acetate will be converted to methane and CO2 in the center of the sludge granule. The research group of Bedstone et al. At the University of Queensland was indeed able to show this layered structure in the granules that were grown on carbohydrate-rich wastewaters, such as brewery effluents. They used fluorescence in situ hybridization or FISH technique to actually show the different trophic groups by different colors. Now in this picture, the acidifying and acetogenic bacteria are indicated in red. The methanogenic archaea converting hydrogen CO2 or acetate into methane in green. And the specific acetoclastic Metanosaeta species splitting acetate into CH4 and CO2 in yellow. The center of this granule apparently consists of unknown mass. A similar study was done with granules from an anaerobic reactor treating cannery wastewater, also containing sugars. Interesting detail here is the close association between the bacteria and hydrogen-atrophic methanogens. Later, Bedstone and his group were able to model the growth and development of the various trophic groups inside anaerobic granular sludge using a kinetic model. Now let's briefly recap the most important issue related to the microbial aspects. First and foremost, the anaerobic microbial conversion process is completely different from that of aerobic ones. Aerobic heterotrophic bacteria have a very wide substrate spectrum. The substrate spectrum of the anaerobes is very narrow and sometimes is restricted to a single compound. The mechanism of COD removal is completely different. Under aerobic conditions, COD is oxidized with oxygen as electron acceptor. In fact, the COD is destructed and the biochemical energy is dissipated as heat and is simply lost. In anaerobic reactors, the COD is converted from complex organic compounds in the most reduced form of carbon, methane, which carries almost all the biochemical energy from the starting compounds. This methane is highly soluble in water and automatically bubbles out of the liquid, so no COD destruction, but COD removal or recovery by methane formation. Anaerobic bacteria hardly derive energy from the mineralization process. Anaerobic digestion is an example of life at a thermodynamic minimum. Moreover, the complex consortia need to share the little energy available in sometimes obligatory centrophic associations. Efficient anaerobic conversion of organic matter requires a close vicinity of all the species involved in the conversion process. The interdependence of the various species in this food web, the need for efficient interspecies hydrogen transfer and the morphological characteristics of the species involved results in the formation of microbial aggregates, biofilms and sludge granules in all anaerobic reactor systems. With this we reach the end of this microbial module. I hope we will meet you in the next lecture. Ciao.